Welcome to the second IGAC-iCACGP ECR online conference in 2025. It's a pleasure to be organizing this event again. My name is Max Desavetaz. I'm a postdoctoral fellow in Australia, and I'm also a member of the ICACGP, the International Conference for Atmospheric Chemistry and Global Pollution. IGAC, for those who don't know, is the International Global Atmospheric Chemistry Project, which was established in 1990. So we are celebrating 35 years of atmospheric chemistry community this year. This early career online conference will probably be happening every other year when the in-person IGAC scientific conference is not happening. So last year, the conference was in person in Kuala Lumpur. And this year, we're once again joining together, bringing this event, which is free to register and submit abstract, to bring all of the early career from around the world without the barrier of having to travel and find funding to attend the event. So we're delighted that you are able to join us today, and hopefully for some of you present your research at the poster session later on. If I can click to the next slide. I've had the chance over the last year to organize this conference with a conference organizing committee. There's 20 plus of us scattered around the world. We've been meeting basically every month or every week in the last month to organize this event. Brought to you the invited speakers, some skills development workshop, and bringing, we hope, an exciting 19-hour of events today. So this is organized in 3 sessions, and we're starting here with the Asia-Oceania time zone. Some context about the IGAC and ICACGP family tree. At the very top, in terms of science governance, is the International Science Council. And underneath this is the International Union for Geodesy and Geophysics. So it's like EGU or AGU you might be familiar with, or their annual meetings. However, the international one stands for the entire world. It's probably less well known, but it is standing above AGU and EGU. Under this are several associations, including IAMAS, the International Association of Meteorology and Atmospheric Sciences. They are one of the sponsors of this event today. And below that, the International Commission on Atmospheric Chemistry and Global Pollution, which I mentioned earlier, was founded just after the Second World War to provide leadership in research for atmospheric chemistry and anything related to air pollution. This is this commission which established IGAC in 1990, and some of the other organizations, we call them sister organizations, that came either out of IGAC or from other communities are SOLAS, which deals with atmosphere and ocean, or ILIPS, which looks more at the ecosystem and atmosphere processes. There is information about IAMAS, ICACGP, IGAC, SOLAS, ILIPS, and also Future Earth, one of the other organizations above us, in the main hall of the conference, and I will come back to this. And Patrick will be introducing the Early Career Steering Committee of IGAC.
Yeah, hello everyone. My name is Patrick. I'm from China. I'm currently a postdoc researcher in Japan, Sophia University. So yeah, I'm a member of the IGAC, ICACGP, ECR, SSC committee since 2023. And this is the current committee members. As you can see, it's a very international group with the members who are from different regions, different countries. Compared with 2 years ago, probably like you can see, we have many new faces actually. And beyond organizing these conferences, we're also hosting like a monthly skills workshop series, which brings together the experts and ECRs to share the practical knowledge. And this is part of our missions to support and connect the global ECR communities. So like, could you next page, please? Yeah. So before we begin, I'd like to share some announcement from the IGAC, ICACGP, ECR, SSC committee. First, we are very excited to announce that we have our first ECR grant call and will be open in next month, actually, October 2025. So This will provide the seed funding to support your research project, conference participations, and other academic activities. And we have a lot of meetings to discuss what we will use of these fundings, and we decided to give you the maximum flexibility to support your own research. So please look out for the details and apply for it. And second, our community will be recruiting new members in 2026. So if you are interested in becoming part of our ECR SSC and helping to shape the future events like this one, please consider applying. And finally, to stay updated on these opportunities, please make sure to sign up for our newsletters and follow us on social media and check our website. You can scan this QR code. If you have time right now, you can make a screenshot. And yeah, lastly, I'm going to announce the skills workshop we're going to host next month. It will from the Dr. Collins to talk about the leveraging open AQ for air qualities, research and applications. And you can scan the QR code on the posters. If you're interested in this workshop, it will give you a very fabulous experience and sharing of his own work, her own work, please. Yeah, that will be this page.
So I'll take it back from there. First, before we really open the schedule of this conference, we would like to thank our sponsors. They're the ones that are making it possible to host the event for free for everybody, and also to have the poster prizes, which I'm sure some of you are very excited about. As a reminder to get you excited for your presentations later, the poster prizes, and there are 6 of them, will be travel, accommodation, and registration fees to attend the 2026 IGAC Scientific Conference in person on the island of Crete of Greece, and attend the ECR short course, which will be happening the few days before the conference. As you can see, we've got some very important sponsors: the World Meteorological Organization, the European Space Agency, IUGG and YAMAS, which I mentioned earlier, MDPI Atmosphere Journal, Future Earth, CSRO Publishing, and the European Atmosphere Monitoring Service, under which the ECMWF model stands. So thank you to all of our sponsors for making this event possible. Another thing, take the time between the sessions and during the breaks to explore the Gather Town. The main hall in particular has been set up in different spaces. IGAC has different working groups which represents different regions of the world where there is research to be done. There's a different working activities on the outside to the top of the main hall. Activities relate more to research topics like urban air atmosphere chemistry, the use of small sensors. B-burn is for biomass burning, and TOR is the ozone atmospheric report. To the lower left corner, you will find the sister organizations like ILIPS, SOLAS, LACES, YES, and ALASIA. And to the bottom right, we have all of our sponsors corner. Around the main entrance where you all appeared on GatherTown, you will also find some information about next year's conference in Greece. Also remember to interact with the different elements in GatherTown. You will have to press the X key on your keyboard when you're near them. And within the plenary and the side meeting room, we have the links to Zoom. So as you walk into the room, you will be offered to click X to join the different Zoom sessions. In terms of schedule, so we are currently at the welcoming and introduction, and we'll move on to our invited speakers in a minute. After that, we have the icebreaker session, the skills workshop with the World Climate Research Program, and then the networking session with some established scientists before moving the joint poster session 1 with the next time zone. So our friends and colleagues from Africa, Europe, and the Middle East will be joining us. During the invited speaker session, don't hesitate to use the Q&A feature in the Zoom webinar at the bottom of your screen. This is how we will be collecting the questions. During the icebreaker and networking session and skills workshop, whenever you're being invited to, remember to turn on your camera and microphone to interact with your fellow attendees. In Gathertown, if you have any issue or have any questions, please contact any members that have organizing committee underneath their name, like is shown on the right-hand side here. And remember, if your poster is being evaluated, you have to be at your poster during the allocated time. The judges have been also told to come and visit you during that time. They have to judge about 2 posters per 20 minutes, so don't panic if you don't see them at the start of your timing. But if by the end of that time you haven't been visited, please contact one of us so that we can check whether the allocated judge is indeed within the room or if we need to reallocate someone to come visit you. If you need certificate of presentation, and that's for the people presenting posters, there are stations all around the poster room where you will be able to request through a form certificate. This form will only open during the poster sessions, so don't panic if you cannot access it outside of the poster session. Just make sure to go and fill in the form while the poster sessions are happening. Finally, just remember to have fun, network with your peers, do exchange LinkedIn if you can, and enjoy this opportunity to learn more. So I will let Emile introduce our invited speakers, and I will just mention that the skills workshop for this time zone is organized and brought to you by the World Climate Research Program Academy, who brings together people in need of workshops or training with those providing them. And Dina and Francis will be introducing that session. And then Dr. Lauren Hoffman will be carrying out, providing the first skills workshop today on the use of AI with climate and atmospheric science. Emile, would you like to introduce the first speaker?
Yeah. Thank you, Max. So, hello everyone. I'm Emile Vargas from the Center for Study of Science, Technology and Policy, India. So I'm here to introduce the speakers for the highlight talk. So we'll be having 3 speakers, and to everyone attending, please feel free to submit your questions in the Q&A panel throughout the talks. We'll be addressing them right after each presentation. Our first speaker is Dr. Emily Franklin, a reactive gases research scientist at the Commonwealth Scientific and Industrial Research Organization, CSIRO, Australia, working in the Atmospheric Composition and Chemistry Group. So today, Dr. Franklin will be presenting Aerosol Pollution in the Modern Megacity, Emerging Sources, Drivers, and Chemistry in a Changing Climate. Dr. Franklin?
Yes, thank you so much for that introduction.
Yes.
And oh, sorry.
Yeah, you may go ahead.
Okay, great, thanks. Thank you so much for that introduction, and thank you to the entire organizing committee for this great setup and for the introduction or the invitation to share some of my recent work, trying to better understand the present and future of aerosol composition and chemistry in the context of a changing climate. And so this work encompasses both work that I'm currently doing at CSIRO and my NSF postdoc fellowship at Colorado State University. So I would like to begin by acknowledging the Merrick people of the Algonquin tribe as the traditional owners of the lands in North America where some of these measurements were conducted, and the Bunurong people as the traditional owners of the lands where the Australian measurements are currently being conducted. And I pay my respects to elders past and present. I would also like to acknowledge and thank all of the people and institutions who have made this work possible, in particular, all of my Frog New York co-authors on our recent paper, as well as the ASPAQ team who have contributed to our measurements that are currently ongoing in Australia. So as we're all very keenly aware, likely in this research community, the climate is changing at a pretty alarming rate with cascading and interconnected effects on the atmosphere. So heat waves are becoming more frequent and more extreme. Wildfires are increasingly blanketing entire regions of the continent in smoke. And in North America, these effects are being seen outside of the sort of Mountain West regions where they've received the most attention in the past decades. Human emissions have also changed, shifting away from vehicle emissions and towards volatile chemical products, whose atmospheric fates are just starting to be explored. And the biosphere, which modulates the majority of organic aerosol production around the globe, is also sensitive to all of these perturbations. So these changes, and in particular, the proposed changes in anthropogenic emission suites, particularly the rising dominance of VCPs, formed a really significant motivation for this work, which is encompassing two campaigns, one in North America in 2023 and one in Australia, which will span mid-2025 through early 2026. The North American campaign, which I'm going to be primarily talking about today, was fluxes of reactive organic gases over New York, or FROG. And this was based at a suburban Long Island site downwind of New York City. And you can see that illustrated in the back trajectory on the left. And so my focus within this campaign was aerosol chemistry and composition, really trying to answer the question of what is aerosol made of, how does it form, and what are the biggest vulnerabilities to various mechanisms of global change. The OSB-AQ campaign is much smaller in scope, encompassing a pared down suite of aerosol chemistry and oxidant measurements. But here I think it's really important to note that Australia and more broadly the entire Southern Hemisphere is really significantly understudied compared to the Northern Hemisphere from an urban aerosol chemistry perspective. And so there's still a lot of room to advance the state of the knowledge in this space from a relatively pared down suite of measurements. So the core measurements underpinning this work are speciated and bulk aerosol composition measurements. So bulk by Aerosol Chemical Speciation Monitor, or ACSM, and speciated measurements by thermal desorption, two-dimensional gas chromatography, coupled with high-resolution time-of-flight mass spectrometry. And so these analyses were supported by measurements of VOCs, aerosol size distribution, oxidants, and local meteorology, as well as being coupled with high split back trajectories, including census population densities, satellite vegetation densities, as well as trajectory heights and meteorology. So from the speciated perspective, the data that we're working with here is really rich and exciting. So using the 2DGC method, we were able to resolve and trace around 900 unique aerosol phase compounds, but it's a pretty challenging data set to work with because most of these compounds can't be identified. So to address this challenge, I developed the CHEMS-RF, or Chemical Characterization by Chromatography Mass Spec Random Forest Modeling, just a bit of a mouthful, model, which is a cheminformatics tool which can predict atmospherically relevant properties of unidentifiable organic compounds based on their mass spectra and their retention times in GC. We next compare these separated compounds to those that are generated in either chamber studies or that are observed in other environments through the UCB Globes database. And finally, through a combination of all these methods, as well as time series clustering, we reach a point where we can apportion our speciated organic material, observe its evolution, and place it in a chemically informative parameter space. So moving on to our major findings from the FROG New York campaign, our measurement period lasted from mid-July to mid-August of 2023. And during this time, the New York region experienced two confirmed smoke influence periods, although notably, neither of these was big enough to garner significant public attention. This was after the big apocalyptic orange skies events of June. And in addition to these sort of smaller smoke influence events, there was also a heat wave in the middle of the summer. So consistent with other sites in this region, the majority of our aerosol material was organic, which is a shift away from the high sulfate aerosol conditions that have been experienced in the past. And through PMF analysis, the organic material was attributed to cooking, hydrocarbons, aka traffic, or hydrocarbon-like organic aerosol, and biomass burning sources, as well as 3 oxidized organic aerosol factors that are more challenging to attribute to a specific source. So when we take a speciated perspective from a numbers basis, volatile chemical products were very prominent, making up about 40% of unique observed species. And of these VCPs, glycols and glycol ethers, fragrances, and personal care products are key contributors. When we combine the bulk and speciated perspective and zoom in on a few key focus periods, we see that during the heat wave, from a bulk perspective, there was a very enhanced dominance of the less oxidized OA factor. And from a speciated perspective, we see very prominent biogenic secondary organic aerosol markers. During one of the biomass burning events, we see enhanced BBOA as well as the MED-OX-OA from a bulk perspective and biomass burning sugar tracers from a speciated perspective. And during a background, relatively low smoke influence period, we see a relatively low biomass burning effect. And from a speciated perspective, we see very prominent VCPs in our background. So altogether, these different methods are allowing us to get some insight into what might be driving the dynamics that we're seeing at our site, with biogenics being dominant during heat waves, sugars from biomass burning being dominant during smoke periods, and VCPs being really significant, particularly in the background. So based on these observations, we've narrowed in on a few key research questions. In particular, what is oxidized organic aerosol? This is a real challenge that faces anybody who does bulk aerosol chemical analysis, as well as a really important question in urban chemistry, particularly under these conditions of climate change, is how sensitive is urban organic aerosol to different mechanisms of global change. So one of our first major findings is that biomass burning is a really significant driver of mass dynamics, despite being a relatively modest fraction of total aerosol. And so what we find is that smoke really sets the baseline of mass variability, while cooking events from local restaurants drive significant excursions above this relationship. And so this is a really strong relationship that's not observed with any of the other factors. And so we really want to understand how this is happening, why it's happening, and what this might mean for how smoke interacts with other sources in an urban environment. So from a mathematical perspective, the reason that these things are so tightly, that total mass and biomass burning are so tightly coupled, is because biomass burning and the OX-OA factors are very tightly correlated. And when we switch over to the speciated tracers, we can see that the reason, the mechanism apparently driving this, is because biogenic secondary organic aerosol is significantly enhanced under conditions of high smoke conditions. And so we've done a bunch of tests to see whether this might be due to covariations from meteorology and other things. And really, there's nothing that can explain it other than the dominance of and dynamics of smoke influence in the region. So about 60% of our OOA variability is associated with biomass burning influence. And the primary mechanism that we're exploring to explain this is that, similarly to how urban plumes in the rainforest drive increased secondary organic aerosol formation from biogenic sources, we believe that we're seeing the same effect with aged smoke over densely forested regions and over cities. Next, moving on to heat waves. When we look at the intersection of temperature and our different factors, we see that on a bulk side, less oxidized organic aerosol is strongly temperature sensitive, with other factors being more complicated. And so when we try and tease out the effects of temperature on our different factors, we see that there are differing levels of contributions from our in our different bulk factors, which together combine to indicate that about 20% of organic aerosol is demonstrating really significant temperature sensitivities. So one caveat to this analysis is that summer 2023 was not particularly hot and quite smoky, so other years would probably have a higher fractional temperature effect compared to some other sources. Something I also want to note here is that population density of where the air mass came from is It's a statistically significant predictor of all of these things, but it's very small in terms of the actual numbers. And this is something that was consistent across our work. We basically didn't see a downwind urban plume effect in the aerosol. In the VOCs, it's different, but we see some sort of confusing dynamics from the aerosol perspective. So zooming back in to the VCPs and trying to better understand what's happening here, since this is such a prescient area of research right now, we observed lots of different individual species, and we see some correlations with air mass population density, which is consistent with the VOC-based literature, as well as sort of other dynamics with air mass history characteristics. However, there's really no connection between any of our bulk aerosol factors, and no VCPs are particularly good tracers of bulk aerosol factors. So from the bulk or ACSM perspective, this really leaves us with the question of, where are they and how are they contributing to things? And so the answer that we came up for this is that basically volatile chemical products don't form a consistent temporal profile. And when they're introduced into our instruments, they don't have prominent mass spectral features, which would indicate that when we're doing bulk analyses, they're there in our oxidized organic aerosol factor, but they're very hard to see. So this question of how important are volatile chemical products versus biogenic organic aerosol is really important to understand the current and future of urban aerosol. So despite all of these challenges, we decided to just give it our best bet with a tracer-based analysis. And so this is assuming that our tracers are small but fractionally consistent drivers of or indicators of what's happening with their underlying source features. And so what we see when we take this analysis and apply it to our different oxidized organic aerosol factors is that there's a good indication that volatile chemical products contribute a consistent background while biogenic SOA drives variability. And just as a sanity check, when we perform the same analysis on the entire mass distribution, what we find is that the fully tracer-based speciated analysis agrees really nicely with the bulk-based analysis pretty much across the board, particularly in terms of the relative importance of biogenics versus volatile chemical products. So what we see here is that when we're trying to better understand all of these different factors contributing to global change, we see as VCPs are contributing about 15%, the biosphere is driving about 40%, fires, both directly and by modulating other things, are contributing about 60%. Heat is contributing about 20%, and combined over 90% of organic aerosol is vulnerable to global change. So this leaves us with lots of interesting remaining questions that are relevant in different areas of the globe. But as I'm running short on time, I'm just going to give a quick overview of our current work in Australia. We've begun measurements in winter that are really showing us some pretty significant pollution events, particularly compared to the very clean background conditions of the Southern Hemisphere. And we're excited for this upcoming summer where we can explore some of these same dynamics in a Southern Hemisphere context. And with that, thank you, and I'd be happy to take any questions.
Thank you, Dr.
very
Franklin.
much. Okay, you go.
Yeah. So we have a question from Vanessa Soon. How did you identify which days were a wildfire smoke event or a heat wave?
Yes, so the heat wave is relatively simple, just based on we used heat index, and also I can say as I was there at the site every single day, and we knew when it was a heat wave because we had to put ice bags on our instruments to keep them running. But so sort of 3 consecutive days when it was a really significant heat wave in the New York region, heat index, I believe, above 41 every day, compared to for the rest of the summer, we didn't have consecutive days above heat index of, I think, 26. In terms of the biomass burning, we used a combination of different methods, but I think very fortunately we were able to partner with a group that was in rural Long Island, and we looked for coincidence of when they also saw these really significant biomass burning tracer enhancement events, because that ruled out any local sources and also confirmed that what we were seeing was sort of continent-wide effects from Canadian wildfire smoke. And we could also confirm this by doing back trajectories and seeing that when the air masses came through that we thought were smoky, they were indeed coming from areas that were on fire in Canada.
Thank you, Dr. Franklin. I have another question. Like, have you compared the mass closure from TOF ACSM to the actual PM1 or PM2.5, and have you observed like what percentage of the organics combined from the ACSM will be equivalent to the PM1?
So are you asking for the speciated versus bulk?
Yes, speciated versus bulk.
Yes, absolutely. So this is, again, pretty challenging because 90% of our individual compounds are unidentifiable. And so I've built a model to attempt to quantify unidentifiable species, but the sensitivity in our instrument can vary pretty significantly depending on functional groups. And so for that reason, it's pretty hard to be highly confident. We estimate it's probably around 15%. And the reason for this is because the sensitivity limitations of the speciated technique are much less oxidized than the bulk, or than we know that what we could see from a speciated perspective is only the relatively less oxidized fraction of the aerosol. However, from a molecular complexity perspective, we expect that this is where the majority of the molecular complexity is going to be, because as things oxidize, they in some respects become a little bit more similar, although they can also diverge, particularly when you're forming larger species. And so this is why we really followed the tracer-based analysis is, for example, our biomass tracers, we know that they were about 10% of total biomass burning, but they're extremely consistent tracers of our bulk biomass burning factor. And so we know that basically the less oxidized things that we can see are really good indicators of what's going on with the more oxidized species that we can't see.
Yeah. Thank you, Dr. Franklin. And
thank you so much.
Yeah. So I think now we can move to the next speaker. Yeah. So our next speaker is Behan Lee. He's currently pursuing his PhD at the Department of Atmospheric and Oceanic Sciences and the Laboratory for Climate and Ocean Atmosphere Studies at Peking University, China. His presentation is titled Discrepancy Between the Model Predicted and Measured Acidity of Suspending Ammonium Sulfate Particles. Behan, the floor is yours. Please go ahead.
Okay, thank you very much, Emil. Yeah. And let me share my screen. So can everyone see it? Okay, very
Yes.
good. So let's start. So hello everyone. I am very honored to be invited here to give this talk. So my name is Weihan Li. I am a PhD student from the Peking University in China. And today the title of my presentation is the discrepancy between the model predicted and the measured acidity of the suspending ammonium sulfate particles. So first is that why we need to care about the pH of aerosol particles. So acidity is one of the most fundamental properties of atmospheric aerosol particles. So this figure is very interesting and it contains a lot of information, but it shows the pH of the aerosol particles and all its relevant atmospheric chemical process. But it's too complicated, so let's only focus on three important information. First is the pH of aerosol particles have a very important influence on both the ecosystem and the human health. Also, it's influenced by some atmospheric process, such as the precipitation and also for the depositions. And second interesting information about this figure is the range of the pH of aerosol particles. So it has a very wide range of like from -1 to around 5, but it's always acidic. It's mostly acidic. The third point is that we also found that the pH of aerosol particles may also be very different from its source. So, and also very different from larger particles, such as the cloud or rain droplets, which means we need to have a specific method to measure the pH of aerosol particles. So why is there this difference? So the reason for the pH difference between the aerosol and its source is mainly because here it shows is the gas particle partitioning of some semi-volatile species like the SO2, CO2, some organic acid, and also ammonia. So there is one very unique semi-volatile species compound among them, which is ammonia. So why is it so unique? So for the other common semi-volatile species like the CO2, SO2, some organic acid, and other compounds, they are mostly acidic. So ammonia is the only one more common basic semi-volatile species that can influence the pH of aerosol particles, which means if the aerosol particles are produced by an acidic source, ammonia is most likely the only gas phase species that can neutralize it. So there is a very important example of ammonium sulfate. So why we choose ammonium sulfate to study is because it's one of the most important composition of the city pollutants. So this figure shows the relationship between the model predicted pH and ammonia evaporation from ammonium sulfate. We found that only 1% of the ammonia evaporation may cause the pH decrease from 5 to 2 predicted by the model. But this is for a model prediction. So the model have a lot of assumptions. They made a lot of assumptions. So are they true? We don't know. Because even for ammonium sulfate, nobody even has really measure the pH of our ammonium sulfate particles. So what is the pH of ammonium sulfate? How does the ammonia influence its pH? So to know this answer, we have to know how can we estimate the pH of aerosol particles. So the existing method can be divided into two categories. The first is to use the thermodynamic models to estimate it. But problem of this method is first, they are models, so they need some experimental result to validate them. So, but if not, how can we know they are correct? Second is that they provide some inconsistent results among each other. So how can we know which one is the correct among different models? Third is that they use a lot of assumptions such as the thermodynamic equilibrium, which may not be the case in the real atmosphere. And second category is the direct measurement. So there are three main existing methods. All of these methods can measure the wide range of pH. And the first two methods use the Raman and the SIRS. And the problem with this method is that they are only applicable to measure the super microparticles. But the most of the suspending particles in real atmosphere, they are submicro, which means they are less than 1 micrometer. And another method is a pH indicator paper method. You collect particle into the pH indicator paper and see its color. It sounds very simple, but it's an offline method since we need to collect them into the pH paper and see the color change. Some black or brown carbons in the air may induce some artifacts for colors. So we need to developed our own method, which is the AFC aerosol fluorescence cell, which you base on the fluorescence. So our method is based on fluorescence. So why can we use the fluorescence to measure pH? I don't know if any of you may have this experience. So when there are some problems with people's eyes, the doctor may drop some green or yellow droplets on the people's eye, and that is the fluorescence dye. So when the fluorescence was induced by the certain wavelengths of light, it emits some light. We emit some fluorescence light. So the doctor can use this fluorescence light to estimate the pH of your eye and see whether there are some problems. So our principle is very similar. So we add some fluorescence dye into the aerosol particles. So the figure shows the excitation spectrum of the fluorescence, and we can change it. It's changed with the pH. So we use two different wavelengths of the laser to excite the particles, and then we can detect the fluorescence. Then the ratio of the fluorescence can be used to estimate pH. So it is that simple. So how can we analyze the data? Before calculate the pH, we have to know the relationship between the ratio of the fluorescence and pH. So we use some box solutions in the small glass cells, some quartz cells. So for the box solution, we already measure the pH by the pH sensor to calibrate this setup. So and then we use an empirical function to fit it. The figure is shown in the left figure. Result is shown in the left figure. So we found that by combining these two different commercial available fluorescent dyes, which is the fluorescent and the Oregon green, we can cover the pH range of from to the 2 to 7, which already covers a large range of the which is very important for the pH of the aerosol particles in the air. So then we use this AFC method and the pH indicator paper to compare with each other. So why we still need to use this pH paper method? As we introduced previously, this is the only one method to measure the submicron particle before hours. So it's like this. So you just collect the particles into the pH paper and see the color shade. So what the result of that is shown in this figure. It is already good enough compared to some previous methods. So the result of the fluorescein or the higher pH range is better, which have only 0.4 units of the difference. But at the lower range of pH for ongoing, the largest difference is 0.6, except for the saturated buffers. But for the saturated buffers, the maximum difference is around 1. So this is because the pH paper was exposed to the air for the photo taking process. So the previous study found that when the pH of the saturated acid droplet can change 1 pH unit for between the around 20% of the RH and all and the higher RH, which is around 90%. So we take this pH paper from our setup to the dry air, so it may change for 1 pH unit, but for saturated buffer we actually don't have other choices on that pH range. So we use our newly developed method to measure the pH of the ammonium sulfate particles. So we made some ammonium sulfate solutions with the fluorescent dyes and use them to generate aerosol particles. And then we mix the particles with the ammonia inside a flow tube. So we control the ammonium concentration and the humidity inside the flow tube. So, and then we measure the ammonium concentration here, pH, using our AFC method, and also the mass concentration, because the total mass concentration of the aerosol particles is also very important in calculating the pH. So they are these are the experimental conditions of our experiment. So we compared our result with the pH indicator paper method, again, because it's the only measurement method except for ours. And we also compared our result with the thermodynamic model, which is EAIM, to investigate the consistency between the measurement and the model. So this is our result. So this figure shows the relationship between the ammonium concentration and pH of ammonium sulfate aerosol particles. So the points are our measurement and the curves were calculated by EAIM. So we found that the result is clearly divided into 2 regimes. So for the high ammonium concentration region, the experiment and model agreed well, but for the low ammonium concentration region, the experiment result is around 3.5 to 4.5, but it's at least 1 pH unit higher than model result, which is 2 to 3.5. But we also found that these 2 different measurement methods agree with each other. So why is it like that? So we think about how to expand this discrepancy. So we made some hypotheses. The first is that the results may be influenced by the addition of the fluorescent dyes, because it's the only one induced change for the composition of the aerosol particles. But we found that the signal becomes stronger with the higher concentration of the dyes, but where pH, which is shown in the points, did not change at all. Then we also considered the uncertainty of the model parameters. We also made our own model to try to investigate it. So one of the most important parameter influencing the gas particle precision is the Henry-Stokes constant, which is this range in the previous study. So we changed it within this range, but the model seems to not change at all. So we found that the uncertainty of the Henry-Stokes constant also cannot explain the discrepancy. And then our third hypothesis is that we found that it may be influenced by some kinetical reasons. So we add 4 big glass flasks after this flow tube to increase the residence time by 1 hour. Then we remain the ammonium concentration to a lower value around this value. And we found that within the 4 hours, the pH of our aerosol particles is still around 4. So why is it like that? So we search for some previous papers, try to investigate the reason and to try to find whether the previous studies has already found the reason. And we found that some previous studies may show that it may take more than several hours for the ammonia to reach the equilibrium in aerosol particles. So which may be the reason for this phenomenon. So the reason of that is that there's a parameter called this alpha, is the mass accommodation coefficient. It means that at the interface of the particle and the gas phase, not all the molecules taken up by the surface can go through the interface. Only a small portion of molecules can go through the surface of the particle. So this fraction, the fraction that the molecule goes through the surface is called alpha. So it can be used to estimate the time for the gas particle partitioning. So this figure shows the result with the pH and this alpha value. So conclusion is that if we found that for to make our result to be possible, only this alpha value should be less than 0.01, means that only 1% of the molecule can go through the interface freely. So is this conclusion true? So we found that also actually contradicts with some of the previous studies. So we search for other explanations. So another effect is about the pH. We found out in some of the previous studies, it's reported that the pH at the surface of the particles is lower than the bulk phase. For example, this study measured the degree of dissociation with the nitric acid. So they find out on the surface, the dissociation of the nitric acid is suppressed, which means it has a lower pH on the surface. So if the interface pH is lower, it can actually explain our results. So the measurement of the pH at the surface of particle is very important for the future studies. So how can we know from this result? We also summarize our results like this figure, and the x-axis is the total sulfate concentration, which are almost totally inside the aerosol particles. And y-axis is the total ammonia, which may be inside the particle or in the gas phase. So when the concentration is above this black solid line, which means the sulfate is totally neutralized by ammonia from the ammonium sulfate, and some extra ammonia, which is ammonia above this line, is the gas phase ammonia. So when the particles are transported from some polluted areas, which may have this concentration, and to the non-polluted areas, some more remote areas, it moves around this direction. And we found that the model predicted the pH will decrease to less than 2, but our results show that pH of aerosol particles may remain for a longer time. So since the lifetime of the aerosol particles are typically longer than the lifetime of the ammonia, it is very important to measure the pH of aerosol particle of the downstream regions in future studies. Another important question of this figure is that previous studies found that the pH of aerosol particles is very important for some solvent formation event for the haze event period. So as you may know, I'm from Beijing. So it's very severe years ago. The haze is very, very severe during the winter time in Beijing. So the main composition of the haze particle is the sulfate. So the right figure shows the formation rate of the sulfate is related with the pH, and also the curve is in different colors indicate the different reaction pathways. What is the major pathways of the sulfate formation during the haze event? It is also very important to measure pH in the real atmospheric particles to answer this very important question. So this is our conclusion. So we have developed our AFC method. It can measure the suspending submicron aerosol particles, and it covers the pH range around 2 to 7. But this range is mainly limited by the for instance dye. So if we have the different it's only for the commercially available fluorescent dyes. If we have more accessible fluorescent dyes in the future, it may expand. So, and also we've also found that the model underestimates the pH of the ammonium sulfate particles, and there are very low ammonium concentration range. And we hypothesize that the equilibrium of the ammonium evaporation from the ammonium particles may not happen within the time scale of hours, and we found that it is very important to measure the pH of aerosol particles in the real atmosphere to further investigate this phenomenon in the future. So thanks everyone for your patience, and this is the list of the reference, and thank you very much.
Thank you, Wehan, for this wonderful talk on how to measure the acidity of aerosols using the offline method. It was really like something new to me because we generally use the models like EIM and Istropia. So I have a question, like suppose you want to measure this, you want to develop this method to measure the pH of ambient particles in real time.
Yes.
How do you think that you can upgrade this?
Yeah. This is also what we think about. So for now, for our experiment in this study, the particles is only we add some fluorescent dyes into the solutions and then generate the particles inside of the laboratory studies. But our final goal is to measure the pH of the aerosol particle in the real atmosphere, right? So I think the major limitation is that how to attach the fluorescent dyes into the particles existing in the air. So I think that is the main goal we need to main barrier we need to of course to achieve the goal to measure the real atmospheric particles. So I think previously there are some studies to attach the fluorescent dyes. There are some methods to attach the fluorescent dyes into the existing particles. And I think by applying some of the previous, for example, I think there is one previous study that they add some fluorescent dyes and then into some solutions and then heat them. So the fluorescent dyes, since they are organic, they may evaporate and then they may absorb by the aerosol particles. So this kind of method may be applicable to our studies. So if we combine them with our studies, then we can attach the fluorescent dyes to the particles in the real atmosphere, and then we can achieve the goal to measure the pH of the real aerosol particles in the real atmosphere, and that is also our goal. So, but for now, this method is still in the early development stage. So we have not done that, but we plan to try that in the future. Yeah.
Yeah. Thank you, Vehan. We have one more question.
Yeah.
Could you how do you take care of fluorescent related aerosol particle, organics or biological in ambient sample? This is a question from Mahendra Chandrajwar from Japan, NES Japan. I can repeat, how you take care of fluorescent related aerosol particles like organics or biological in the ambient sample?
Yeah, I think that is a very critical question in that if we want to apply this to some ambient samples. I think what we always think about that the one of the limitation of our method is that we always assume that all the aerosol particles are internally mixed and there are no phase separation in the particle itself. But in the real atmosphere, there are some organic particles and also the phase of the particle may separate. So the particle itself may separate to the organic phase and the aqueous phase. And if that happens, the fluorescent dye may go to the organic phase for all of them. So that is maybe the one limitation if you want to apply it to the real atmosphere in the future. But we also search for it and we also think about the possibilities. So we also found that in the previous studies, some of the previous studies used the fluorescent dye to try to investigate the phase separation, and also there are some fluorescent dyes that is sensitive to both the phase separation, the phase of the both the organic and also the pH of aerosol particles, and that may be used for the that may use for our future studies, and also that there may be some of the aerosol particles, organic aerosol particle itself have the fluorescence effect. So that is also very important. I think we may need to try to find a way to separate that fluorescence and our fluorescence from fluorescence dyes, and we may need some spectrum studies. But that is also for the future. So in our study, for now we don't use any spectra, but in future the to analyze the spectrum, the fluorescence may also be important if we want to extend this method. So thank you very much.
Yeah, thank you, Vehan, once again. So now we'll move to the next speaker. Our final speaker for this session is, thank you. Our final speaker for this session is Dr. Pratibha Vishwakarma. She recently completed her PhD in Environmental Engineering from IIT Kanpur, India, with a focus on the chemical characterization and source apportionment of pollutants. And today she'll present on the seasonal distribution and source attribution of dicarboxylic acids in fine atmospheric aerosols over the Brahmaputra Valley, northeastern India. Dr. Vishwakarma, over to you.
Hello. Am I audible?
Yes, yeah.
Okay. Thank you, Emil. And hello, everyone. I am Dr. Pratibha Vishwakarma, and today I am here to present my topic on source apportionment and seasonal distribution of dicarboxylic acids. So let me share my screen. Yeah. So my screen visible? Is anyone can confirm me? Is my screen visible?
It is visible, but not full screen yet.
Okay. Okay. So
you please put it in the slideshow?
Yeah, it is on slideshow. Okay.
Yeah.
Thank you. Okay, so the seasonal distribution and source of distribution of dicarboxylic acids in fine atmospheric aerosol over the Brahmaputra Valley, northeast India. Today, I will discuss on this topic. And so first, we should have some little bit information about aerosols, that what are the aerosols and how they are important, and in the atmosphere from where they are coming. So for the quick introduction, that I will discuss that aerosols are the assembly of the solid and liquid particles which are suspended in gaseous medium, which are long enough to be observed and measured, and generally they are from 1 nanometer to 100 micrometer. As here we can see that, let me show my pointer. Okay, so here we can see the size distribution of the aerosol and how they are penetrating in our respiratory system, at what reason. So they are the matter of the concern because they are very hazardous for health, and also they have adverse effect on the climate. So aerosol can be natural or anthropogenic both. So they can be in the atmosphere from the natural aerosol like fog or mist, dust, forest institute, and the geyser system. And also they came from the anthropogenic activities like smoke and other burning activities like biomass burning and fossil fuel burning. So here we can see that the size of the aerosol matters because on the basis of their size, it can penetrate different regions of the respiratory system. So it is also it also has adverse effect on health. And if we talk about the climate, so how they affect our climate, and I'm talking about the cloud formation and rain. So let's have a little bit idea of this. So let's if we have a small cesium concentration in the atmosphere, so it will have it will uptake the water on, and condensation will happen, and there is a lower number of the large droplets, so it is the normal condition. But if we have highest concentration of the cesium in the atmosphere and it take water onto that, so what happens? So there will be a small droplet in high number, and also there's a longer lifetime which doesn't precipitate, and they are more reflective and have higher albedo. So there will be a two condition. First condition is no rain condition, and when these accumulation become very high, and then now the cloud cannot hold that much droplet, then there's a condition of the catastrophic rain, which leads to the flood-like situation. So the concentration of the aerosol into the atmosphere in a very high amount is not good for the cloud form for the rain perspective. So if in India, there is a project like National Carbonation Aerosols Program, in which we have 11 sampling sites and 18 institutes involved in this. So the purpose of this project is we need to find the seasonal distribution of the carbonaceous aerosol, and we identify and quantify the sources of the anthropogenic aerosol and carbonaceous constituted over the India, and also what are the impact of the aerosol on the regional climate. So these are the key objectives of this project, and IIT Kanpur has been involved in this project, and now we are assessing 2 major sites of the India with respect to the formation and the effect of the aerosol into that region. So what we have done, we have targeted some compound which is known as the organic molecular markers. So what are these compounds? These are the tracer compound which can help to identify the particulate emission sources, and with the detection of these compounds, we know from where they are coming into the atmosphere and leading to the formation of the aerosol and affecting the climate and the health of the people. So I have also worked on the PHS, polycyclic aromatic hydrocarbons, but here I will discuss about the dicarboxylic acids, which is one of the molecular markers. And so dicarboxylic acid in the atmosphere can come from the 2 emission, like primary emission and also the secondary formation. So like if you talk about the primary emission, so biomass burning, vehicular emission, and the biofuel burning, these are the most common like primary emission of the dicarboxylic acids. And also there is a large part of the photochemical oxidation in the atmosphere which lead to the formation of the diacids into the atmosphere. And so here I have targeted C3 to C10 in this study. I have also targeted C2, but it was not that much detectable in the project study. So I have only calculated from C3 to C2 high dicarboxylic acids. And here we can see the structure of the DCAs. So there are some like some processes of formation of DCAs, like if we talk about CN, azelaic acid, so it can come into the atmosphere by the oxidation of the biogenic unsaturated fatty acid, so like linoleic acid or oleic acid. And also like C3 and C2, they come into the atmosphere by the direct emission of the from the primary sources. So there are different possible pathways of the formation of these diacids and their presence in the atmosphere. So now here in this study, I am talking about the Brahmaputra Valley. So this is my study site, which is called Jorhat. This is situated in Assam in a Brahmaputra Valley. And so to know the possible sources of the particular compound in the atmosphere, first we need to know what are the important geographical locations in that vicinity. So now here we can see that this is the my location in the northeast India, and the location name is Jorhat. And if we talk about the geographical main points in this study, so there is a main city in the 5 kilometer away northeast direction, and also the industrial estate in 6 kilometer away in the southeast direction. Also, there are many small and medium enterprises, industries in this area, which are like tea processing industries. These are the possible local sources of the diacids into the atmosphere. And so to detect the dicarboxylic acid into the atmosphere, first we develop optimized method of the detection of the DCA. So what we have done, we have collected this sample at our study site with the use of air sample, and then on the PM 2.5 filter. And then we go for the sample extraction using the SAM instrument for the sample extraction. And we use here methanol as a solvent. And then we go for the rotatory evaporator for the evaporation. And then we have done derivatization using the BSD-FA 1% TMCS. And after that, the chemical correction has been done using the GCMS, gas chromatography mass spectrometry. So this method was developed to detect the DCA, which has a lot of trials and tests involved in it. And after that, with good extraction efficiency, we were able to quantify the DCA into the sample. So after developing the protocol for the detection of the DCA, and then we go for the detection of the sample for the 1 year long sample for the Jorhat. Now we have here shown what are the effect of the meteorological parameter on the DCA concentration. So here in this red line showing the DCA concentration, and this green is showing the PM 2.5 concentration. In this study, we have only quantified the PM 2.5 bound DCAs. And this is the meteorological parameter like relative humidity and temperature and precipitation. So now I have categorized the whole season in 4 parts, like winter, summer, monsoon, and post-monsoon. So here we can see that the DCA concentration is highest in the winter, like it is also getting high as compared to other seasons. But here PM2.5 concentration is not high as compared to winter. So the possible reason of the getting high concentration of DCAs in monsoon and not having high concentration at the same time of the PM2.5 is might be because of the formation of DCA by the secondary transformation, like photochemical oxidation and other chemical reaction. So, or also there's a precipitation also. So maybe that the PM particles get precipitated due to the high precipitation here as compared to other seasons. So now I have included this whole DCA concentration with the wind direction for the local sources identification. So this is the pollution dose diagram of the DCA, where here I have included the wind direction with wind direction and the DCA concentration to get this pollution dose diagram. So as I have discussed earlier that in northeast direction there's a densely populated city, and in this southeast direction there's a industrial state, including the tea processing industries, and also the Jorhat airport in this side. And there are several small gaon in these 8 villages in this area. So we can say that these are the possible local sources in for the particular compound at this particular site. So now this is the normal seasonal distribution of the DCA in different respective seasons like winter, summer, monsoon, and post-monsoon. So here we can see that we are getting all like C3 to C10, all carbon DCAs in the winter. But in monsoon, we are not getting, and in post-monsoon, we are also not getting the lower carbon DCA. So there is lots of atmospheric chemistry involved here. So like in winter, we are not getting the favorable condition for the atmospheric reaction, photochemical oxidation, and other chemical transformation as compared to monsoon and post-monsoon. So maybe it might be the reason that in winter we are getting a particulate bound DCA in winter in all like carbon like C3 to C10, but we are not getting lower carbon change DCAs in the monsoon as compared to other seasons. So there are other reasons of the possible finding of the DC into the atmosphere, which I will discuss later. So this is the this is the slide showing the what are the possible pathways of the formation of the DC into the atmosphere. And here I have shown the correlation of the DCS among the DCS in different seasons. So here we can see that C8 and C10 are getting good correlation in winter and monsoon season, and also the C3 to C4 having good correlation in winter season. So the having good correlation means that there may be possibility of the DCS coming from the same sources, or the coming from the same sources, or getting the and getting oxidized from the one source from the one DCA to another DCA. So these are the possible pathway of the formation of the DC into the atmosphere. That like if we say that C8 and C8, C9, C10 getting good correlation, so there may be possibility they are coming from the same source, or maybe that C9 get oxidized to C8. So that's why they are having good correlation. And if we look into the OC-EC ratio, that is organic carbon elemental carbon ratio, and the potassium ion and EC ratio. So what this ratio indicates, if we are getting high ratio of the organic carbon, elemental carbon into the atmosphere, like approx to 2 or more than 2, so the tendency of the photochemical oxidation is high as compared to the direct emission. And as we know that potassium like is a ratio of the biomass burning. So potassium ion on EC ratio is if high, so biomass burning, the dominance of the biomass burning is high over the fossil fuel emission. So for the present side, we are getting higher OC by EC ratio in monsoon season as compared to winter and other season. So we can say that the dominance of the photochemical oxidation for the formation of the DCA is high as compared to other season over the direct emission. So if we talk about the presence of the DCA in monsoon, so we can say that photochemical oxidation is predominant pathway for the formation of DCA. And here we are also getting the biomass burning. Biomass burning is a dominated factor So we are getting VOCs from the biomass burning, which are get photo-oxidated into the DCA formation into the monsoon season. And if you talk about the winter season, so we can say that the fossil fuel burning dominated over the biomass burning, and the most prominent sources of the DCA into the atmosphere are the direct emission from the anthropogenic sources. So this is the whole atmospheric chemistry behind the DCA. Like we can say that cyclohexane, which is coming from the anthropogenic activities, get oxidized into adipic acid, and then adipic acid oxidized into glutaric acid. So there are other marine fulvic acid, like if we have linoleic or linoleic acid, like high unsaturated fatty acid, which get oxidized using the oxidant like H2O and hydroxyl radical. So these are getting oxidized into marine fulvic acid, and then it further oxidized to azelaic acid. So this is this is coming from the marine or terrestrial plant, unsaturated fatty acid, and the cyclohexane, which is coming from the anthropogenic activity. Similarly, here we are seeing that isoprene and monoterpenes, these are coming from the biogenic sources and get oxidized into the DCA into the atmosphere. So this is the possible pathways of the DCAs into the atmosphere, which we are getting into at our study site. So now for the study site Jorhat, we have now run the high split bacteriometry to know from where they are coming to the long range transport at the study site. So what we have done a high split model, and now we see that in winter the long range transport is coming from the western and and also the some middle part of India. So and if we talk about the summer, so the air mass is coming from the western and also the Bay of Bengal sites, and having we are getting here the long range transport at the study site. But in monsoon, that only that Arabian Sea and Bay of Bengal we are getting air masses from these these sites. And in post monsoon, like most prominent, most dominated long range transport, like the nearby areas. Like we are saying that in post monsoon, like we are not getting like western or Arabians or Bay of Bengal. We are getting more of the air masses from the nearby area like Assam and Nagaland, and some of the one trajectory here from the middle of India. So now this is the only direction for the direction of the long air masses coming from different side at the study location. So further, what we have done, we have combined this air mass back trajectory with the concentration weighted trajectory method. So here what we have done, we have calculated the weighted concentration for the each grid cell which are contributing towards the DC at the study site. So here what we have done, we have assign the we have used the residence time of the trajectory and concentration of the each sample, and then we have calculated the CWT value, concentrated weighted trajectory value, for the each possible sources region. So what we are getting here, we are getting the CWT values, which are showing the concentrated weighted carboxylic acids value for the each grid cell, which are showing the what are the extent of the DCS coming at my location. So if we are saying that the high red patches or dark patches, which are having high CWT value, contributing more DCAs at the study site as compared to other seasons. So if we are seeing in winter we are getting high value of the DCAs from the like Arunachal Pradesh, Nagaland, and Mamama as compared to other regions. And similarly for summer, like we are getting high DCA emission regions, which are Assam, Manipur, and Arunachal Pradesh. So these are the, and here like there will be the possible reason, like in winter season, we are getting higher CWT value among all the seasons because of the maybe less because of the atmospheric condition, we are having lower boundary layer height at the study site, and also that there is no vigorous reaction in the atmosphere, and also that trap of the pollutant within the boundary layer, which are not which is not get diffused according to atmosphere. So the highest symptom of a winter season, it is justified with the meteorological parameter reasons. And also if we talk about the monsoon and post monsoon, so here we are getting that that Assam and Meghalaya and Manipur and Middle Indian City and the Bay of Bengal. Also, they are the most possible reasons for the contribution of DCA in the monsoon season. And also in post monsoon, similarly, we are getting Assam and Arunachal Pradesh, which is having high CWT value in the post monsoon season. So these are the this is the concentration weighted trajectory model for the DCA at the study site. And if we conclude this whole study, so what we have done, we have calculated the DCA concentration starting from in all seasons, and we get that monsoon has the highest concentration of the DCA and summer has the lowest, and then second most highest concentration in winter. And what are the possible sources into the atmosphere into the of the DCA is the direct emission from the anthropogenic activities, and also and in some seasons there is a predominance of the photochemical oxidation of the biogenic precursors like from the from vegetation or byproduct of the animals, and also the terrestrial plants and from the agricultural plants. So we are getting both kind of biogenic precursor and also the direct emission of the DCA into the atmosphere. And also we have done here for the short range for the local sources, we have done that DCA pollution dose. And also the long range transport, we have done air mass back trajectory, which is showing the northwest and the southwest of the study area, middle Indian region.
Sorry to interrupt. Could you like finish in 1 minute? We are a little bit short of time.
Okay. I'm about it. This is the last slide. Okay. So middle India region and Bay of Bengal are the possible reasons for the long range transport of the DCA at the study site. So these are the differences which I have used in the study for this slide. And thank you for your attention. Thank you.
Thank you, Dr. Vishwakarma. Can you hear me?
Yes.
Yeah, okay. Yeah, so now we'll move to the questions. We have two questions from Ramya C. Balan. Her first question is, what fraction of DCAs and associated compounds are present in the total aerosol concentration ambient aerosol? And the second question is, what is the source for C8?
What is the source for?
C8.
Okay.
C8, C10.
So,
yeah.
Okay. So as I have discussed that C8 mostly sources like C8 is from the anthropogenic activities. Like if we are getting like cyclohexane and other hydrocarbons from the anthropogenic activities, so they get photo-oxidized into the atmosphere and then they form the C8 into the atmosphere. So also and what is the other question?
The other question is, what fraction of DCAs and associated compounds are present in the total aerosol concentration in ambient aerosols?
Okay, so here we have total aerosol concentration. Okay, so in this study, we have only quantified the PM 2.5 DCAs. So we have done GCMS analysis. So which is we have like prepared a protocol for the DCA detection. So whatever concentration we are getting here, which is only PM 2.5 DCA. Like if we talk about the organic content, if we talk about the organic carbon in the total PM 2.5 mass, so it is about like 70 to 80% for the total PM 2.5 like mass. So, but for the DCA, like whatever we are PM 2.5 DCA, like it is like related to the organic carbon. So we can say that like 70 to 80% like for all our PM 2.5 mass is like DCAs.
Okay, yeah, thank you. We have another question from Jasmita Khadki. For high split, what spatial resolution of the meteorological data is used? Was arbitrary weight functions applied for CWT?
Okay, so for high split, we have used like planetary boundary layer height, which is for different like we have done analysis for season wise, like winter, summer, monsoon, and post monsoon. So we have taken the average planetary boundary layer height and the 5 days back trajectory, which is like total runtime of 120 hours. So this is like data I have inputted and also with the latitude longitude of for my site, and also what the orbital weight function applied. Okay, so in the CWT analysis, we have just given the concentration for the each day and the resident and also the with the high split data, which is used like which the system used for the calculator residence time. So according to like how much resident time each trajectory spent in a particular grid cell, with the like if I have shown the equation that the concentration of the particular trajectory into the resident time spent by that trajectory upon the total resident time for all trajectory in that particular grid cell is used to calculate the CWT value.
Okay, yeah. Thank you, Dr. Vishwakarma. Yeah, I think that's it. And once again, thank you to all the speakers for staying on time and delivering such engaging presentations. And all of you and all of the attendees for your thoughtful questions and participation.